DNA is made up of phosphates, sugars, and bases. The bonds between two bases are called hydrogen bonds. The bonds between sugars and phosphates and sugars and bases are called covalent bonds. Adenine and guanine are purines, while thymine and cytosine are pyrimidines. Sugar in DNA is called deoxyribose. Helicase unwinds the DNA and breaks the hydrogen bonds separating the complementary base pairs. The single-strand binding proteins hold the sides of the DNA apart. Complementary bases form a new half of the DNA, controlled by the enzymes DNA polymerase. It is only read from the 3' to the 5'. RNA primase attaches to the start. DNA polymerase will begin its work after primase tells it where to go. The leading strand is easier to read because it starts copying from the 3' to the 5'. The lagging strand is harder because you have to start at the helicase and then go work towards the end. These sections on the lagging strand are called Okazaki fragments. Each fragment has about 1,000 to 2,000 base pairs each. Okazaki fragments are only created on the lagging strand. DNA polymerase goes back over the RNA primase to make sure everything is okay. Finally, ligase reconnects all the bonds to form two complete DNA molecules.
Step 1 of transcription. RNA polymerase attaches to the DNA molecule at the promoter, the start signal, TA. Step 2. Complementary bases form a new mRNA molecule. Step 3. RNA polymerase reaches the termination signal, the stop signal. Step 4. The DNA rewinds and rezips.
How is it awkward not talking?
Step 5. Certain parts of the pre-mRNA are removed. Introns leave and exons stay. Poly-AAA tail is now added.
Step 1 in translation. mRNA attaches to a ribosome. First, tRNA ribosome attaches to the start codon AUG and makes a amino acid. Step 2. The next tRNA joins with mRNA according to the codon on mRNA. The amino acid joins the peptide chain by forming a peptide bond and eventually a protein. Step 3. The used tRNA is released into the cytoplasm. The next tRNA is positioned, the polypeptide chain grows. Step 4. Termination. The process ends when a stop codon is reached. Step 5. Disassembly. The ribosome falls apart and the protein is released.